we will get started. Good morning, everybody. Welcome to our weekly Grand Rounds. Before we begin and I hand it over, I would like to respectfully acknowledge that we are located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to enrich our vibrant community. Before I hand it over to Dr. Ritchie to introduce the Henry Pabst lecture, just a reminder that we do start daylight savings time on Saturday night, so remember to fall back. Over to you, Bruce.
Thanks. It's a great honor, actually, to introduce the Henry Pabst Lectureship, which has been going on for 3 or 4 years now. It's in honor of Henry Pabst, who is a pediatric immunologist in Edmonton and was a mentor to many, many, many people. Can I show the slides? Are these the slides from?
Yeah, the next one. I have a photo.
You have to be smart. Oh, there we go. Henry actually was born in Germany and grew up during the Second World War, coming to Canada sort of as a refugee after the war, and ended up in Edmonton really as a young man with no support, and built a great life and a career here. He was a tremendous mentor to many, many people. He was a great clinician with an interest in immunology and genetics. He was famous really locally for looking after children with complex genetic problems at the Glenrose Hospital, where his goal was really to keep these kids out of ICU. And he was very successful at it by paying attention to all the details. So a great clinician, an engaging scientist, and really a wonderful, kind man. I worked with him on an immunodeficiency treatment program with subcutaneous immunoglobulin back in the early 2000s, and he provided all the patients. I got all the recognition, and he didn't care. But afterwards, we set up a program with this new treatment, and I was setting up a bunch of programs at the same time, and he just slotted in and looked after the whole immunodeficiency program for a long time before he got sick with Parkinson's disease. So a wonderful man. I won't tell you all the stories about him, but there are a multitude of really interesting stories, both medically and scientifically. It's great. I know Mo was going to introduce Stu, but I had a good chance to talk with Stu last night at talk, and it struck me that he is really a great representative of the things that Henry believed in immunology and genetics and good clinical care. So without further ado, I'll hand it over to Mo.
All right. Thank you very much, Bruce. Yeah, there we go. Okay, good. So thank you all for coming today. It's great to see people in person. So on behalf of the clinical immunologists in the Department of Medicine and Pediatrics, because some of them are here, I would like to actually welcome you and thank you for coming to Grand Rounds. It is really my pleasure to introduce Dr. Stuart Turvey, who really is a great example of an impactful translational scientist. I mean, I'm a translational scientist, so when I was going through his CV, I have to say this was very humbling. So Dr. Turvey, his research is really focused on explaining how clinical observations can lead to molecular discoveries, which can in turn be utilized to guide diagnosis and also better therapy, which I think is kind of a nice synopsis. Is that fair? So Dr. Turvey he's a professor of pediatrics at the University of British Columbia. He completed his Bachelor's of Medicine and Surgery with first-class honors at the University of Sydney. And then he went on to complete a Doctor of Philosophy at the University of Oxford. He's a Rhodes Scholar. And then subsequently, he came back and was recruited to the University of British Columbia and has been decorated with lots of awards and has done tremendous work. So just to mention a few awards, he has received the CSACI, Canadian Society of Allergy and Clinical Immunology Award for Excellence in Research, and he is recognized as a Fellow of Clinical Immunology Society Designation Award, which is very, very prestigious in this field. So he's a clinician scientist, and he holds a Tier 1 Canada Research Chair in Pediatric Precision Health and the Aubrey J. Tingle Award in Immunology. In addition to this, he has published 481 articles as per my search this morning with an H index of 64. He has mentored many students, many junior colleagues, and many immunologists across the country, I think. So with this, I would like to ask you to thank Dr. Turvey for joining us, and welcome, Dr. Turvey.
Thanks. It's a great honor, actually, to introduce the Henry Pabst Lectureship, which has been going on for 3 or 4 years now. It's in honor of Henry Pabst, who is a pediatric immunologist in Edmonton and was a mentor to many, many, many people. Can I show the slides? Are these the slides from?
Yeah, the next one. I have a photo.
You have to be smart. Oh, there we go. Henry actually was born in Germany and grew up during the Second World War, coming to Canada sort of as a refugee after the war, and ended up in Edmonton really as a young man with no support, and built a great life and a career here. He was a tremendous mentor to many, many people. He was a great clinician with an interest in immunology and genetics. He was famous really locally for looking after children with complex genetic problems at the Glenrose Hospital, where his goal was really to keep these kids out of ICU. And he was very successful at it by paying attention to all the details. So a great clinician, an engaging scientist, and really a wonderful, kind man. I worked with him on an immunodeficiency treatment program with subcutaneous immunoglobulin back in the early 2000s, and he provided all the patients. I got all the recognition, and he didn't care. But afterwards, we set up a program with this new treatment, and I was setting up a bunch of programs at the same time, and he just slotted in and looked after the whole immunodeficiency program for a long time before he got sick with Parkinson's disease. So a wonderful man. I won't tell you all the stories about him, but there are a multitude of really interesting stories, both medically and scientifically. It's great. I know Mo was going to introduce Stu, but I had a good chance to talk with Stu last night at talk, and it struck me that he is really a great representative of the things that Henry believed in immunology and genetics and good clinical care. So without further ado, I'll hand it over to Mo.
All right. Thank you very much, Bruce. Yeah, there we go. Okay, good. So thank you all for coming today. It's great to see people in person. So on behalf of the clinical immunologists in the Department of Medicine and Pediatrics, because some of them are here, I would like to actually welcome you and thank you for coming to Grand Rounds. It is really my pleasure to introduce Dr. Stuart Turvey, Stu, who really is a great example of an impactful translational scientist. I mean, I'm a translational scientist, so when I was going through his CV, I have to say this was very humbling. So Dr. Turvey's research is really focused on explaining how clinical observations can lead to molecular discoveries, which can in turn be utilized to guide diagnosis and also better therapy, which I think is kind of a nice synopsis. Is that fair? So Dr. Turvey he's a professor of pediatrics at the University of British Columbia. He completed his Bachelor's of Medicine and Surgery with first-class honors at the University of Sydney. And then he went on to complete a Doctor of Philosophy at the University of Oxford. He's a Rhodes Scholar. And then subsequently, he came back and was recruited to the University of British Columbia and has been decorated with lots of awards and has done tremendous work. So just to mention a few awards, he has received the CSACS, the Canadian Society of Allergy and Clinical Immunology Award for Excellence in Research, and he is recognized as a Fellow of Clinical Immunology Society Designation Award, which is very, very prestigious in this field. So he's a clinician scientist, and he holds a Tier 1 Canada Research Chair in Pediatric Precision Health and the Aubrey J. Tingle Award in Immunology. In addition to this, he has published 481 articles as per my search this morning with an H index of 64. He has mentored many students, many junior colleagues, and many immunologists across the country, I think. So with this, I would like to ask you to thank Dr. Turvey for joining us, and welcome, Dr. Turvey.
So embarrassed. Thank you for that very kind introduction. I want to say two things. So to follow on the land acknowledgement, so I'm based in Vancouver at BC Children's Hospital. So we're located on the traditional unceded territories of the Musqueam, Tsleil-Waututh, and Squamish nations, and that's where we do our work. So we do acknowledge that land. Also, I did my homework about Dr. Pabst and was impressed by this man. And the theme that came through, I think, was his humanity and his commitment to his patients. And I feel like we as clinicians never go wrong if we keep our patients at the center of our universe. And I had the sense that this was exactly where his center was. So this is me. I'm a pediatrician. So you know, the center of my universe is patients who come to see us and patients who come to our hospitals. And so the research we do really is embedded in trying to improve health outcomes for children. But more broadly, and what I'll talk about clearly spills into adult medicine and beyond. And so we're going to really take you on a journey that started in BC Children's Hospital, but led to the discovery of a new human disease that causes severe allergies. And I think the reason why that story is relevant for us is beyond defining a new disease. I think it reflects something that we can do within our Canadian healthcare system. So with our healthcare system, we ultimately funnel patients to tertiary centers who will have unusual, maybe brand new diseases, whether in pediatrics or adult medicine. And it just sort of challenge us now to harness the technologies we can use to recognize, diagnose, and transform outcomes for those patients. And so what we're talking about really is rare diseases. And so from a pediatric perspective, rare diseases really matter in children's hospitals. So we estimate that in children's hospital, about 1 in 3 children admitted to a pediatric hospital has a form of rare disease. You know, less in adult hospital, but clearly these people move and become adult patients as well. And for many of these rare diseases, we don't have diagnoses yet. Patients and families who live with rare disease go through a diagnostic odyssey of test after painful test, physician visit after physician visit, looking for answers. And often they don't get the one because they don't have a name for this disease yet. And why this matters today is that, you know, we have the opportunity to utilize sequencing technology to make those diagnoses and then to transform care for these patients. And so that's really at the heart what precision medicine is. And I think this is helpful to realize that medicine is transformed by technology. Technology changes the work we do, whether it's, I don't know, X-rays or the ability to do blood counts or any form of technology you might like. And then, so what's transformed us in the field of rare diseases has been the availability of next generation sequencing. So exome sequencing or next generation sequencing was first applied clinically in 2010. And we've seen an explosion of diagnoses of single gene defects driven by this technology, where now it's really overtaken what we do. And then for us as clinical immunologists, this is exciting times, and we've seen a similar explosion in these diseases. And so people look at this thing and sort of date themselves and wonder what diseases were known when I was around. So, 1972, adenosine deaminase deficiency, the gene was described. That's really the first one. 1992, if you're looking at this list, was the gene for X-linked skid, or the bubble boy disease. I started my faculty position at UBC in 2004. I had to know about 100 diseases as a trainee. Now our trainees are supposed to know more than 500, I guess. The truth is we don't actually need to know these diseases individually. We need to know that we can access technology to make those diagnoses. And what we as clinical immunologists typically are looking at are what we call inborn errors of immunity, or primary immunodeficiencies. So these are genetically determined disorders of the immune system. They mean that parts of the immune system are missing or dysfunctional. And so patients come to see us with infections, autoimmunity, malignancy, and other evidence of immune dysregulation. These are not so rare. And I think that's, so when we have each of these 500 or more diseases are individually rare, but when we aggregate them, we're seeing prevalence numbers of perhaps at 1 in 1,000, 1 in 10,000. So our practices and our practice rosters will include these patients, many of whom are potentially unrecognized and undiagnosed. And so making that diagnosis is transformative. We can offer remarkable therapies now. We can offer accurate genetic counseling for the families to understand what the risk might be for subsequent pregnancies or for other family members. And these patients who have single gene defects really provide us with amazing insight into human biology. And I don't know if there's fans of mouse knockout models here. We built immunology on mouse knockouts from the 1980s onwards, but now we can start to actually, study these individual humans to understand how the gene functions in the intact human system. The field of primary immune deficiencies has really emphasized infections, and that's what we've seen. So here I'm showing you the Jeffrey Modell Foundation. It's a charity committed to awareness for primary immune deficiencies, and they're 10 warning signs that they put out there. They're all about infections. Here on the right is David Vedder. This is the bubble boy who was born with no T cells, who grew up until his teenage years in this bubble to protect him from infections. And that's sort of what I think we recognize. But the truth is that that's really just the tip of the iceberg. And so when the immune system doesn't work, our patients will also have malignancy, autoimmunity. And what I want to emphasize today, which is perhaps the least appreciated, is this idea of allergic disease. So unusual and severe allergic disease. And so the group of diseases we're talking about here are called the primary atopic disorders. This was a term coined in 2018 by John Lyons and Josh Milner, looking to describe a subgroup of patients with severe single gene defects that caused allergies. And some you'll recognize, hyper IgE syndrome or Job syndrome, maybe the classic, Wiskott-Aldrich syndrome. But there's a growing list of now 30 to 50 of these conditions. And so our group's been sort of interested in really trying to raise awareness of those and identifying clinical red flags that might drive us to consider a primary atopic disorder. And so what we emphasize now is the patients will have atopy or allergies. They'll have that allergic manifestation. They'll have the biomarkers we recognize of significant allergic disease with the high IgE levels, the high eosinophils. But beyond that, they'll also tend to have other features that we should recognize. There can be short stature. Kids that don't grow well, adults that have short stature is an important part. Connective tissue disorders, so pathologic fractures, normal structure of the face, various forms of autoimmunity, increased chances of malignancy, and then some will have the infections, but it's not really the sort of fundamental driving part. And so I think the message I'd hope people will take away from from this today is think about those patients that you might see and just think perhaps we should be working them up for an underlying genetic immune system disorder. And I think my allergy colleagues have traditionally not really thought so much about single gene defects, and I think adult practice physicians who care predominantly for adults often don't think of single gene defects. But we're now realizing that there's actually a very good number of these patients, and it will really transform the lives by making those diagnoses. Okay, so that's what I wanted to talk about and encourage you is to think about genes, and we'll talk towards the end about strategies that we might use to make these diagnoses. But before that, I just want to tell you a story. And this was a story that started in BC Children's Hospital. It probably was in the Starbucks lineup. I don't exactly remember where the conversation happened, but speaking to a senior physician, his name's Michael Sear. He was a pediatric respiratory doctor who, I think every hospital has these doctors who he's just retired, but he saw thousands of patients, was an astute clinician and could remember them all, remember their name, remember them. And he came up to me in the coffee lineup and said, Turvey, you study these rare diseases where children have problems with their immune system. You need to go and track down this young man. He for sure has one of your problems. He's probably 30 or 35 now. Go and find him. I'm sure he'll have a problem and he'll have something really unusual. So I listened, because what I've come to realize is that the number one predictor of having these types of diseases is exactly that story. And it's kind of unquantifiable, but it's experienced clinicians who say this patient really doesn't fit out the typical disease course. And so I'm talking about immune system problems, but this I think holds true across our disciplines. It's the outlier who doesn't follow a disease course that we really should be thinking broadly about these types of conditions. And so here's the story of this young man that started the journey. So he was the only child of unrelated Caucasian parents from British Columbia, really had no notable family history. He got sick very soon after birth. So within the sort of first month of life, he developed widespread, significant atopic dermatitis that was difficult to treat and unresponsive to typical therapies. Over time, as foods were introduced, he had challenges with feeding and ended up seeing gastroenterologists and was found to have eosinophilic gastrointestinal disease, so eosinophilic esophagitis, but also beyond. This was a huge issue for him in terms of eating. He had multiple food allergies, ended up with a very restricted diet, kind of meat and potatoes diet. He had asthma that would flare in the context of viral infections, and he'd end up in hospital. He didn't grow well, and I'll show you his growth charts in the next figure. He had some infections, but they're really the infections that we'd expect to see with disrupted skin barrier, so cutaneous infections with bacteria, fungal, candida, and molluscum. So, but not deep-seated invasive infections that might have made people think about immune system problems. So this, I'm sort of summarizing a 35-year course, but diligent physicians treated him with, the newest therapies that moved along, but ultimately, really the only thing that gave him any kind of comfort and disease control were oral corticosteroids, and he could never, wean his steroid dose below about 10 milligrams of prednisone a day, and so he had accumulated morbidity related to that. He had very high eosinophil counts and high serum IgE, and to sort of fast forward this story, we enrolled him, we reached out to him, we were able to enroll him in a research study. When we'd felt that we knew what he had, we actually called to say, hey, I think we know what you have, we wanted to share this. And we learned that the week before he died of a cerebral hemorrhage. which is probably actually part of his disease as well. And I think disruption of the cerebral vasculature is part of this new disease that he had. So we never got to share or tell this young man what he had or give him closure on that. Here's his growth charts. Pediatricians love growth charts. And so here we're showing his weight across age from 2 to 19. Here's his height. And you can see, you know, his growth, both height and weight, were often below the 3rd percentile. So that for us as pediatricians meets the criteria for failure to thrive. So this is really how we kind of approach these patients with our translational research program. So clinicians will come to us with these types of patients. Things have moved, and so now more and more often the patients have had some form of sequencing, and the clinicians may now have a genetic change that they think might be the cause of the disease, but we need to do work in the lab to really interrogate and prove that. So we'll do functional validation in the research lab to understand that. We also are very committed to treatment. It's not enough for us to just diagnose. And so in the lab, we're able to test potential treatment options to really close that loop. And then we do a lot of reaching out and looking for other patients. And so we'll reach out to kind of the global immunology community through, for example, we have a listserv, sort of email service for Clinical Immunology Society that we can write emails and say, hey, we found this thing. Does anyone have them? So we did that. We posted the initial experience on a preprint server with the intent to find more patients. And then there's actually a sort of formal matching thing called Gene Matcher, which is basically an online registration where you can enter your gene and say, hey, and if someone else has entered patient with their gene, you'll get an email to say, hey, you should talk. So like, I don't know what it's like, but it matches you based on your genes, and you can have a conversation about patient phenotypes and whether that might work. So we sort of followed this cycle that started with that one coffee shop conversation and ended up describing a brand new human disease in the last year or so. And so what I want to tell you about now is what this young man has and what now a good number of patients have, which is germline gain of function variants in a gene called STAT6 that are the cause of this severe allergic disease. So you can't have an immunology talk without some immunology signaling cascades. So there won't be that much of this, but we just can't do it without it. This is in our soul, I think. So STAT6 lies at the heart of allergic inflammation. Okay, that's what you really need to know. And so the major drivers of allergic inflammation are these Th2 cytokines, IL-4 and IL-13. They signal through their appropriate receptors, activate the JAK molecules that have become much more recognized with a growing number of JAK inhibitors. And then that activates STAT6. STAT6 becomes phosphorylated, dimerizes, goes to the nucleus where it's a transcription factor and drives pro-allergic gene transcription. This protein, this transcription factor, lies at the heart of allergic inflammation. Knockout mice that lack STAT6 don't get allergic disease, essentially. And we can look at that another way, that IL-4 and IL-13 that signal through STAT6 are really the drivers of those various aspects of allergic disease that we recognize, whether it's the high IgE and the anaphylaxis, whether it's the changes in the airway related to asthma, whether it's changes in the skin, the high eosinophils. So this protein, this gene, was a fantastic candidate to drive allergic disease. Beyond that, we had more than a decade of genome-wide association studies showing that more common genetic changes in STAT6 were associated with kind of all the sort of various versions of allergic disease that we could measure, whether it was asthma or, eosinophils and other things. And so, we knew that this was a fantastic gene, a very strong candidate to go after. And what happened was, I told you that we started with the one young man in British Columbia. Through kind of medical genetics networks, we identified a second patient in Ontario through our colleagues at McMaster University. And at that stage, with these two, is when we started looking for others and posting the preprint servers. And I think testament to the cooperative nature of the clinical immunology community, we went from these 2 Canadian cases to a consortium that spanned here, Vancouver, across to Hong Kong with Iran and Europe. And we did that, I don't know, in 2-3 months. Like, it happened remarkably fast, and was, I don't know, just a huge privilege and honor to link with these committed clinicians across the world who really had astutely identified patients with this problem, but they didn't have others. They couldn't really be sure. And here's the group we put together with that consortium. So we ended up with a group of 16 patients, which for us in rare immunology is actually a lot, and across these 10 families and across 3 continents. And I'm showing you the pedigrees here, I think, to give another important message. And so some of these families had the classic inheritance patterns that I learned about in medical school, right? We ask about family history, and for example, here in family J, there was these multiple affected family members across generations with different sexes, and so this was typical of an autosomal dominant inheritance pattern. And we see that again in a couple of other families following this inheritance pattern. But what I think sticks out to me from this, and what's a very important message for us clinically, is that about half of these were sporadic cases, no family history. So here, for example, family G, this is actually the young man from British Columbia. Healthy parents, the parents didn't carry this genetic change. And we'll get into the genetics later. And he developed it as a de novo spontaneous change. And we saw that read out time and time again in these families. And so that's a message that I think is growing with increased access to sequencing, is that we don't need a family history to have a single gene defect. So don't let that put you off. If you think that this patient is an outlier and has really unusual disease, don't be put off by the lack of family history. If you have the family history, don't ignore it, but don't close your eyes if there's no family history. And when we put all of these patients together, they look very much like that first young man I described. Early onset severe atopic dermatitis, multiple food allergies that led to restricted diets, recurrent respiratory tract infections, predominantly viruses that would trigger asthma exacerbations, the skin disease that I explained. Many of them had eosinophilic gastrointestinal disease that was often beyond the, I guess, more common eosinophilic esophagitis. They had the biomarkers we expect, and 2 of these individuals, 2 of the 16, had died of problems that were, I think, likely related to the underlying disease. One of anaphylaxis after NSAID exposure, and the other of the cerebral hemorrhage. I'll just kind of show you some pictures to put this in context. So this is what the skin looks like in these patients. So this was, bad skin disease. The biopsies showed that chronic lichenification and eosinophilic infiltration. This was what the esophagus looked like. So it had the features of EOE with the longitudinal fissures and the trachealization, those horizontal bands, and again, visible high numbers of eosinophils. These are the biomarkers. So I put the IgE and the EOs actually on a log scale. We tend to use different units sometimes for IgE, but I'm showing you what the normal range here is in gray. So we had very high eosinophil count, and And sort of relevant for this audience, we see that our oldest patient was out into their 50s. So these were not just children. And the eosinophil counts, ran high. This dotted line is at 1.5 or 1500, that sort of hypereosinophilia number. The gray bar is 0.5 or 500, where we would sort of define high eosinophils. So they were typically high. Some responded to treatments, but they were typically high. So that's biomarkers that we would think about. The problem is, and this is deliberately small because you don't have to see it, all the other things that we might look at in the clinic really didn't give us any hints for these things. Maybe the platelets and white count ran high in some of them, sign of perhaps chronic inflammation. But all those other standard clinical measures we might use, or flow cytometry numbers of T cells and B cells and natural killer cells and other things, didn't tell us. There was no kind of typical biomarker for these diseases. So we needed to go to genetics to get answers for these patients. Okay. Now I'll tell you a little bit about the genes and sort of take you through the process of how we work this out. So these patients didn't all have exactly the same genetic change, but they had genetic changes in STAT6 that were functionally the same. And I'll show you that evidence. So when we do whole exome sequencing, whole genome sequencing, or even panels, one of the things we'll do first is to see if that genetic change is seen in large population databases. So here we're using the large population database called NOMAD and looking at the allele frequency. And the patient's variants in red were not seen in any of these now several 100,000 healthy individuals. So these were very rare variants. And then we can apply bioinformatic tools to try and predict if these might be harmful or disruptive variants. And one common score is called this CAD score. And what we can see is that the patient variants ended up with quite high CAD scores. There's various ways to measure this, and were very rare. So this was evidence that these might be functionally relevant. We then did what we might do, is to think about where do these variants lie within the protein structure. So here's the sort of linear view of the STAT6 protein. And what we could see is that here we're denoting where the patient variants were. And quite quickly, it's clear there's something going on. This amino acid, so amino acid position 419, lots of the patients seem to be hitting this specific amino acid. But we also saw them in other parts. DNA binding domain made some good sense in terms of that, but they were spread out. But we then were able to use the known 3D structure. And so what I'm showing you here is, this is the DNA double helix sitting in the middle. And this is then the 3D structure of the STAT6. And we're highlighting where those genetic changes are. And, they're not randomly distributed through the protein. They're not sort of out here in these tails. They're all right at the interface where the protein, the STAT6, talks to the DNA. And that's what transcription factors do. They go find DNA and activate transcription. So this sort of made some good sense biologically that these may affect the way STAT6 works. The other bioinformatic tool we were able to leverage was called COSMIC. So COSMIC is the catalog of somatic mutations in cancer. And so huge global efforts to sequence cancers, to understand what those genetic changes are, those somatic genetic changes are that drive disease. And we know that many of those somatic variants will be gain of function, giving the cancer cells a survival advantage. And so when we looked at that, this is what you come out with from STAT6. And I've just sort of color-coded the ones we saw in our patients. But remarkably, we saw all these 419 variants in our patient population, and then 419 is the hotspot for somatic variants in STAT6. So this is it, right? This is a tower of recurrent somatic mutations in that. And then a good number of our other variants that we saw were also here in the COSMIC database. So giving us some more, I guess, circumstantial evidence that these might be gain of functions. This sort of raises the question that you might be thinking, well, you know, what's the lymphoma risk? What's the cancer risk for these individuals? And, you know, the answer is we don't truly know yet. This is a kind of brand new disease with small numbers, but I'll tell you this anecdote that, you know, is concerning, I think. So this is a family that had a genetic change at position 419, mother, 2 affected sons, 1 who died of anaphylaxis. And this mother is the oldest person in the cohort. She was diagnosed with B-cell lymphoma around age 40, was treated, and then 10 years later relapsed with a more aggressive form of B-cell lymphoma. And so she, in her germline, carries one of these very common driving mutations. So it's an anecdote of one, but I think thinking hard about cancer risk and particularly lymphoma risk in these patients makes good sense. And so we then had this information. So we went to the lab to look to prove, are these genetic changes really functionally relevant? What do they do? And so we used a assay called luciferase assay, where we could look at how well that STAT6 promoter was activated. And we hypothesized that those patient variants would be more turned on than the regular variants. And so here I'm showing you that. Here we look at wild type variants or some other controls. And when we stimulate, for example, the wild type with IL-4, it goes up quite nicely. But when you look at all those patient variants, they have a high baseline that's, again, on a log scale is fairly maxed out, but goes up somewhat. And so we're seeing that these variants were all much more turned on. We did that another way, looking at phosphorylation of STAT6, and the answer was very much the same, that the patient variants here in red were much more turned on than the wild type variants. Again, giving us cellular evidence suggesting there might be a gain of function. If you're really into biochemistry, we measured it other ways. And so what we concluded from this series of cellular models was that we felt that we had evidence now that the patient's cells were much more turned on, that there was a fire burning in terms of activation of STAT6. But we weren't satisfied just with the cell models, and so we went to our patients and to our collaborators and wanted to look in the primary organ, the blood cells of these patients, to see what was going on. And here's one of the first experiments we did. So these global collaborations are amazing, but you have to sort of get cells across continents and borders, and so getting them fresh and in good shape was not straightforward. And so the cells came, and I was breathing down the neck of my PhD student saying, you know, it's going to be amazing. We'll stimulate the patient cells and they'll like turn on way more and we'll have solved this problem. And so he did this experiment, and here's what happened. They stimulated the patient cells and they were exactly the same as and I said, oh, you know, do it again. And it was always the same. And I was sad. And so we shared this with some of our collaborators, and there's a kind of famous person in this field called John O'Shea from the NIH. And he said, you know, you got to remember that turning off the immune system is equally important as turning it on. And it's really about and if it turns off differently, that may be explaining this exaggerated response. And so we said, sure, you know, let's do that experiment. And lo and behold, at least and we have more data supporting this, it seemed to be right, that when we stimulated, they were about the same, but when we took that stimulus away and washed away the IL-4 and looked at the resolution, the patient cells here in red did that much more slowly. And so the area under the curve of immune activation was increased. And we've shown that many more times now. And then we measured other markers, other biomarkers of allergic inflammation in those patients, and showed that the patients had high levels of of cells that were making some of these classic cytokines that drive allergies, drive eosinophilia, so IL-13, IL-4, IL-5. And so it, I think, started to generate a nice package of data. So I told you that we really want to treat patients, and making diagnoses is nice, but let's treat them. And what's exciting for all of us now is that big armamentarium of medicines we have that's growing, that we can borrow from our oncology colleagues or rheumatology colleagues or whomever else, and repurpose for our patients. And so as we looked at that biology of exaggerated IL-4, IL-13 signaling going through this receptor, activating JAKs and STATs, some clear candidates emerged that we should try. So one was dupilumab, monoclonal antibody against the alpha chain of the IL-4 receptor. That's common for both those cytokines. That was a good candidate, clinically approved, even down to 6 months in kids for bad allergic disease now. Relatively available as well. And then JAK inhibitors as well, again, approved, understood, and relatively available. And so we then did some work in the lab to check 3 of these. We looked at dupilumab and then 2 JAK inhibitors, ruxolitinib and tofacitinib, and checked them in the lab to see what happened. And here in the wild type, we showed that, yeah, all those medicines, as expected, would turn down that increased IL-4 signal. And again, in a range of these patient samples, they also decreased that IL-4 signaling. If you're really into this, you might notice something that I think is interesting, is that the JAK inhibitors were able to decrease the baseline signaling in a way that the dupilumab didn't. And that makes sense because of the mechanism of action. And so we shared this with our sort of global cohort, and then the colleagues in Hong Kong came back and said, well, you know, we've been doing everything for that young man with the severe skin, the photo I showed you. And we actually put him on dupilumab a while ago, and it's been amazing. And so here I'm showing you now his response to dupilumab. I showed you his skin, and then you know, much improved. His and then various measures of skin disease, the SCORAD or EASY, showed this really nice improvement. And then I'll show you my very favorite figure. This is it. Told you pediatricians like growth charts. So this is the young man in Hong Kong, and this is a bad growth chart. Here's the 3rd percentile, and you can see that he was so far below that for his height and his weight. His physicians were diligent, had tried everything, had given him growth hormone here when he was about 16. His growth velocity didn't change at all. And then at 19, he started dupilumab, and he grew 20 centimeters in a year, and put on, I don't know what he put on, 30 kilos or something like. And I met this young man, and he told me that it just changed his life. He was in this dark place of illness and disease and felt there was no hope. And now he had hope. He could go and work. He could function. So I know that's why I get up in the morning, I guess, was just these stories. So it was remarkable. And we saw this played out in other patients. Interestingly, in some jurisdictions, they couldn't access dupilumab, but could access tofacitinib. And so the patient in Iran, his story was also remarkable. So he had terrible eosinophilic esophagitis. He was a teenager, and every month they would have to dilate his esophagus so that he could swallow. And he started tofacitinib, and after about 6 weeks of that, he didn't need to go and have his esophagus dilated anymore. And again, really just changed his outlook. So I think, you know, we'll see what medicines are the best, and we'll understand the options. But by making this diagnosis, we're able to repurpose some of these medicines and make dramatic changes. So So to conclude this bit of the story, we identified from starting with just a single patient and a conversation in a coffee shop, which is, a conversation will likely happen in this institution today. We identified a brand new human disease. We're showing some of those mechanisms of what's going on. We haven't defined it all yet, but we're some of the way there. We have identified some effective treatments, and we want to acknowledge that the full phenotype of this condition will require recognition of more patients. And what starts with kind of a single patient really can change things. And so this is interesting in that as we moved forward on our story and published our story, actually 3 other reports of different families also came out around the same time, so with exactly the same problem, and in fact some of these same genetic changes. A family in Bangkok, a family in Tokyo, and a family in Istanbul with exactly this disease. And so for us, that's very gratifying. So now I just want to end with like 3 slides. So I've told you kind of a long story, but I want to sort of bring that a little more practical about what we could do, today in our Canadian healthcare system and how we might work up these patients. And so before we get into it, I think it's sort of just helpful to kind of have a little refresher on what our genome is and what's there, so that we understand the limitations of the sequencing technology we have. So human genome is sort of made up of introns and exons. And as we move through making mRNA, we splice up those introns and end up together. All those yellow exons move together. And that's what we call, when we aggregate those, we call that the exome. And when we look at that, the whole genome has about 3 million nucleotides. Each of these patients had 1 nucleotide of those 3 million spelt wrong, and that's the cause of their disease, which is always, it's kind of amazing to me. But that's and then the exome makes about 1 or 2% of the whole genome. And so just sort of appreciate what we're sequencing when we say we've done genetics clinically. The other bit to remember is there's a lot we don't know yet. And so the estimate is there's about 20,000 genes in the human genome, but only 5,000 so far of those have been linked to human disease. It's likely, I think, that all 20,000 have the potential and will cause human disease. And so, again, sort of a very clinically focused look that only looks at known diseases, potentially misses, 3/4 of those genes. And so, what genetic test do we send? Obviously, that depends on our specialty, depends on our healthcare system, depends on what we get approval for. But in general, these are the options that are there today. So most common would be gene panels of our diseases that we know, and they might range from tens to perhaps a couple of hundred genes of known disease-causing genes. So the immunology panels that are available clinically today don't have STAT6 on them yet. They will sometime soon, but they don't. They're really good. They have great coverage and give us good information, but only look at what they look at. Exome is a really powerful study that will identify both known and unknown diseases, but has some higher costs. There's also the potential for finding incidental findings, so genetic changes unrelated to the reason we do the sequencing. And so pre-test genetic counseling is very important for that. And then whole genome sequencing, I think, is difficult to access clinically for most of us, is the most comprehensive, but we don't really understand yet all what's normal human genetic background change. And so it's computationally difficult to interpret. But so these are the tests we have, but realize that we haven't done genetics if we do a panel. There's many more genes. We haven't done genetics if we do an exome, because we're missing 98% of the genome. And even if we've done a whole genome, we don't always understand what's going on. So I guess my message is think about genetic diseases, but if the tests are coming back negative and you really in your heart feel that there must be something going on, revisit that, as this technology is changing and advancing so quickly that we should sort of revisit that and revisit with our medical genetics colleagues to keep asking the question, because we're not done with the test that we can potentially access. And so with that, I will end. And here is a list of 95 people who contributed to that large study. And I really want to acknowledge the first author here. His name is Mehul Sharma. He is a PhD student who tackled this project. And then the long list, I think, really acknowledges the generosity of the clinical immunology community around the world. So with that, I will end, and thank you for your attention.
So we're going to take questions online first, and then perhaps we'll take questions from the audience. So I have a question from Paige. So Paige is wondering if people have considered using the dupilumab, which is a TSLP1 inhibitor, for treating steps in skin function.
Might be me. No, I mean, not. I mean, again, it's a brand new disease, and when we've talked to our colleagues, so dupilumab is the one we had some of this anecdotal evidence on, and has been the one that most of these patients have tried. But I think, yeah, exactly. We have an open mind, and I think that medicine, may be really effective as well. So I agree with that for sure.
Excellent. Anybody have questions here? So Jan Willem and then Norman. Sorry.
So thank you very much. So interestingly, the gain of STAT6, these patients developed lots of hemophilia, but also infections, and those are very good. You talked a little bit about malignancy, but not about autoimmunity in this disease.
Yeah.
So it's a very good background for what we call EGPA. Staph infections in combination with these classic hemophilia. So were there patients already who had MPO ANCA or did develop vasculitis?
Yeah. So I think the question is, were the people who had vasculitis had their ANCA positive? So yeah, no is the interesting answer, but it's a tiny. I want to acknowledge this is a tiny cohort. And now that we're starting to sort of understand these diseases, we are following them more closely with our colleagues, and people have been generous with that. But yeah, you're absolutely right that autoimmunity hasn't been a big and obvious feature of this, but I think it's also about how hard people have looked for those things.
Index patients did have cerebral.
Yeah, and so I could talk to that. So you're right, and I didn't get into this, but now sort of single gene defects in a variety of those proteins have been found, and so STAT1 gain of function or Job syndrome, JAK3 sort of dominant interference are also known to be associated with vascular aneurysms, cerebrovascular aneurysms. So this patient died of the cerebral hemorrhage, and a number of the other patients have had brain MRIs, and we haven't seen aneurysms, but what seems to be there actually like multiple rare structural anomalies sort of within the circle of Willis and things. So I think there's a lot, these are germline variants that I think are actually influencing development as these proteins function, in development in different ways. So again, I worry about that, but it's N of one experience.
No.
Thanks. What a fantastic talk. I think it really gets us sort of thinking about those rare birds that we see, and getting people like Mo involved in those patients and so on. As a non-geneticist and non-immunologist, I just was curious with that variant, the genetic variant you showed in stat 6. Is there any sort of, coming back to precision medicine and what you just talked about, is there any correlation with severity of disease depending on where the variant is, or response to therapy, more importantly, right?
Yeah, no, that's where so the patients were recognized by their astute clinicians based on this sort of clinical phenotype. But you're absolutely right that when we dug into that, there was, clinical variation. That patient in Hanoi was a young girl with bad but not terrible skin, but had been on a ventilator in the ICU 5 or 6 times before her 3rd birthday. So she had a different phenotype. I didn't want to bury you in the science, but we've done, for example, looking at transcriptomics or looking at kind of the RNA profiles, and each genetic variant or each family's variant has some different patterns of disease, and we're looking to try and understand that. In terms of response to therapy, dupilumab and the JAK inhibitors have proved to be pretty effective, probably because they sort of target that sort of central fundamental parts. But yeah, the scientists in us now are really into looking at those things like, do they bind more strongly to the DNA? Do they change the way the genes are moving? And I think the answer will be yes, but we don't know yet.
Yeah.
Yeah, I have another treatment. Thank you so much for that.
Yeah.
A treatment-related question, kind of related to that gain of function mutations in general. If you lived in a magical world and you had access to all the therapeutics, do you think the money is treating the JAK or downstream cytokines? Like, what would you want?
I mean, they're sort of, I think they're self-fulfilling cycles. And so interestingly, the problem is at the stat, which was kind of the bottom of that cascade, but I showed you that dupilumab, which binds the receptor, or the JAK inhibitors work because that transcription factor then drives, for example, IL-4, IL-13 production, also drives upregulation of that IL-4 alpha receptor. So I think because they are these kind of self-reinforcing cycles, we do okay targeting sort of wherever it is. I like the appealing thing of the JAK inhibitors is that they seem better at turning down that baseline kind of grinding inflammation that I think is going on, and then oral medicines with pretty good safety profile is appealing. Dupilumab is also a good medicine. So right now when people ask, I will say, whatever you can access within your healthcare system is reasonable.
Okay, so there's time for one question, so I will ask the question. So one of the striking things I noticed is that these patients did not develop fibrosis. And yet we know that IL-13 is very pro-fibrotic, and so is activation of SAT4 and some of the other stats. Why do you think this happens?
Yeah, I don't know. I mean, I think now that the patients are sort of understood and have an answer, we can, with our colleagues, go and ask these questions to look for, I don't know, like pulmonary fibrosis, for example. The colleagues didn't recognize, sort of identify that as a problem. This cohort, runs on the younger side, obviously. So we've been encouraging them to look at these sort of questions and look more broadly to define the phenotype properly. What was kind of obvious was, terrible skin, can't eat much, and that was, with the terrible skin, can't sleep and can't function. And that was what the physicians was seeing. But now I think as we control some of that, we can ask other questions and look more deeply.
I think it's really cool because it's a somatic mutation that's going to function.
Yeah.
So you can access things like fibroblasts from the skin.
Easily.
So this is a very nice and helpful answer. Thank you very much.
Well, thank you. I appreciate it.
Okay.